بسم الله الرحمن الرحيم. السلام عليكم ورحمة الله وبركاته. طلبة المرحلة الثالثة، وأهلا بكم في محاضرة جديدة. Our lecture today is about practical pathology, about the cell injury. As you know, the normal structure and function اللي حكينا عنه يعني previous lectures about normal structure of cells and the cellular components and compartments. مثل ما تعرفون يعني as introduction للcell injury قلنا احنا يعني should be know the normal structure and function of cells. As you know, the cell have cellular different type of cellular organelles, and these cellular organelles, each of one has a specific function that should be performed within normal structural limits, and any defect in these organelles may reflect on the whole structure and function of the cell, resulting in cell injury. And the cell injury means cell disease or diseased cell يعني study of the disease at the level of cell. مثل ما قلنا انه المودرن باثولوجي هو يعتبر انه الديزيز start from the يعني at the level of cells and even at the level of cellular compartment. يعني وين ماكو defect at the level of cellular compartment, this will reflect on total structure and function of the cells, and then on the organs and on the system, and manifested as clinical signs and symptoms on the whole body. Okay. Cell function مثل ما قلنا انه maintain the production of energy and maintain the normal physiological and environmental يعني between the internal environment of cell and external environment, to maintain the normal homeostasis via different cellular compartment. The main compartment cellular component is a plasma membrane إنه هو maintain يعني constant and concentration of internal ionic gradient, ويعتبر هو selective permeable. Admit allow the certain molecules to enter the cells and excluding or extruding other unwanted molecules. It provides structural envelope and contain informational and synthetic and catabolic constituent. Also it is a provide environmental milieu for signal transduction of molecules. Any defect in cell membrane will result in defect in one or all of these plasma membrane function. Cell injury including two types: reversible injury and irreversible injury. Reversible injury, for example, cellular swelling, including cloudy swelling or hydropic degeneration, and fatty change or fatty degeneration or steatosis. مرات نسميه steatosis. And this type of injury is curable or reversible. Other type is irreversible injury, which include apoptosis, either physiological apoptosis or pathological apoptosis, والnecrosis هو pathological only. This means cell death or irreversible injury. Causes of cell injury: either genetic causes or inherited causes. يعني from parents to transport through genetic material into their progeny. Or acquired causes. The acquired causes include hypoxia, ischemia. Ischemia means blood deprivation or a decrease of blood supply for certain area of tissue or organs. Physical agent, for example, radiation and trauma. Chemical agent, strong alkaline or strong acids. Microbial agent such as bacteria, viruses, fungi, parasite. Immunological agent, for example, the hypersensitivities or autoimmune diseases. Nutritional causes such as يعني hyponutrition or hypernutrition, starvation. Psychological causes, also the psychological state considered as one cause of cell injury. This is unknown or non-cell causes, but there is unknown causes for certain diseases such as idiopathic muscular atrophy. يعني ممكن انه الكوز ما نعرفه او يسمونه الايديوباثيك unknown or idiopathic. for particular يعني vulnerable system يعني الحساس or sensitive systems in the cells هي cell membrane integrity والadenosine triphosphate generation, protein synthesis. طبعا cell membrane integrity have critical function to maintaining the ionic concentration and gradient and maintain the normal osmotic homeostasis for cells. Adenosine triphosphate ATP generation. If there is any defect or decrease in ATP production, this reflects the underlying defect of mitochondria and aerobic respiration or oxidative phosphorylation. لأنه oxidative phosphorylation depend on oxygen occur in mitochondria and responsible to production of ATP. Synthesis of protein performed by ribosomes on rough endoplasmic reticulum. إذا there is any defect or change in rough endoplasmic reticulum or ribosomes, مثلا مرات يصير لها detachment from rough endoplasmic reticulum راح يصير decrease or defect in protein synthesis or protein misfolding. The integrity of genetic material or genetic apparatus of the DNA. Ultrastructural change of reversible injury. Ultrastructural change means the change that only can seen under or via the electron microscope. لأنه كل هاي changes at the level of cells and cell يعني component and compartment cellular organs اللي يصير بها change cannot be seen under the light microscope. They are only can be seen by electron microscope. لذلك يسمونها الالترا structural changes. البلازما ممبران يصير ب flipping and distortion microvilli, loosening and of intercellular attachment. طبعا البلازما ممبران يصير defect or loss of its selective permeability. Mitochondrial defect and swelling resulting in decrease in ATP production, dilation of endoplasmic reticulum, particularly rough endoplasmic reticulum, and detachment of ribosomes, with a decrease in protein synthesis, nuclear alteration, clumping of chromatin, يعني genetic material, reversible injury. Two pattern of morphological changes correlated to reversible injury can be recognized under the light microscope. These are cellular swelling, including hydrophobic or cloudy degeneration or swelling, and the fatty changes or steatosis or fatty degeneration. Pathogenesis of reversible injury. Pathogenesis, for example, loss of ATP generation by mitochondria, initially resulting in reversible events. Loss of sodium potassium, normal sodium potassium ATPase membrane pump, lead to loss of ionic and osmotic gradient of cells. فراح يصير increase sodium and water influx into the cells, and increase efflux of potassium due to decrease of ATP. Also increase calcium influx into the cells associated with the sodium, and the water also associated with sodium influx into the cells, and the cells will be water overloaded with water, in association with decrease in potassium concentration, concentration in cells. يعني يصير عندنا potassium efflux of the cells, resulting in swelling, cellular swelling, and endoplasmic reticulum dilation or swelling. Also increase anaerobic glycolysis, anaerobic glycolysis, resulting in glycogen depletion and lactate or lactic acid accumulation, and this will lead to decrease in pH and converting the internal environment of cell into acidic internal environment of cells. This the acidity of cells, in association or in combination with increased concentration of calcium, this will lead to activation of lysosomal enzymes, hydrolysis enzymes, which result in cell lysis or autolysis, cell autolysis. Reduced protein synthesis due to ribosomal detachment from rough endoplasmic reticulum. This figure show the normal cell structure and reversible injury of cell and irreversible injury. The normal cells, if under any stress or injurious stimuli, will undergo a reversible change of cells, total cells, including cellular component such as cell membrane, here the cell plasma membrane, flipping of plasma membrane, and generalized swelling of the cells, clumping of chromatin inside the nuclei, and autophagy of unwanted or defected mitochondria, also aggregation of intramembranous particles, endoplasmic reticulum swelling, dispersion of ribosomes or detachment of ribosomes from rough endoplasmic reticulum, and this will lead to decreased protein synthesis, mitochondrial swelling, and deposition of small densities, and this will contain high phospholipid amorphous densities, phospholipid amorphous densities inside the mitochondria. At this stage, the cell can return to normal state if the injurious stimulus or the causes is removed. The cells can be returned to normal state, normal physiological structure and function. But if the causes or injurious stimuli increased or convert to severe stress on the cells, exceeding the ability of cell, or maybe continue for a long time and progress, the cell may undergo irreversible injury and cell death, affecting all cellular structure. For example, rupture of lysosomes and start the cell autolysis, nucleus become pyknotic, and then undergo karyorrhexis, and then karyolysis, means complete lysis of genetic material, lysis of rough endoplasmic reticulum and defect of destruction of plasma membrane, large deposition of amorphous densities, and severe swelling of mitochondria and rupture, and this will lead to cell death. Cellular swelling. The commonest and earliest form of cell injury include cloud swelling or degeneration, hydrophobic degeneration, vascular degeneration. The gross finding of this change: the organ that affected by this change appear paler in color, increased turgor, means swelled or enlarged, and increase in weight. Microscopic finding: the nucleus is not appeared under that microscope لأنه ultra structural، يعني nuclear change cannot be seen under that microscope. مثل clumping or detachment only in the end stage، يعني karyorrhexis, karyolysis, complete karyolysis can be seen under that microscope. Increased hydration or water overload of cells resulting in hydrophobic degeneration, or cell swelling may be associated with accumulation of coarse granules in the cytoplasm, and this refers to cloudy swelling, or maybe accumulation of pigment cast or protein or hyaline cast resulting in hyaline degeneration. Hydrophobic degeneration. The hydrophobic degeneration appear here in a hepatic field. This is typical example of hydrophobic degeneration, represented by severe hepatocyte swelling and the border of cells or plasma membrane appear with thin and swelled, and the nuclei nearly in the center, and the cytoplasm appear clear due to water overload compared with normal hepatocytes, compared with surrounding normal hepatocytes, and the cells appear different in size. Some of swelled cells appear large, and some medium size appear with medium size and small. But all this focus of hepatocyte undergo prominent hydrophobic degeneration. This figure show or illustrate other example of hydrophobic degeneration in myometrium muscle, smooth muscle of uterus. Also, the smooth muscle cells appear swelled, and the cytoplasm clear, and the nuclei located in the center of nuclei surrounding with clear or empty space due to water overload. This refers to or example of hydrophobic degeneration. This is typical example of cloudy swelling in kidney, where here the glomeruli undergo with severe cloudy swelling and surrounded with renal tubules, and the renal tubular epithelium appear with a prominent cloudy swelling, where the cytoplasm of renal tubular epithelium appear with turbid cytoplasm, and the nuclei clear in the center. This turbidity of cytoplasm due to the accumulation of coarse granules in the cytoplasm of renal tubules. This is a typical example of cloudy swelling. Fatty degeneration or fatty change or steatosis. It is abnormal fat storage in non-fatty cells, due to mismatching between supply of and demand for fat in the cells, and also due to defect in fat metabolism in the cells. Gross feature of affected organ appear as enlarged and become yellow in color due to excessive fat accumulation, and soft and smooth and soft and greasy in appearance. This is a gross feature of fatty changed affected organ, or fatty changed organ. Microscopic change. The cells fatty changed appear as clear small vacuoles inside the cells. Microscopically, fatty chains appear at the level of cells as clear small vacuoles, multiple clear small vacuoles يسمونها المايكروفيزيكولار ستيتوسيس. شوف multiple microvesicular or micro vacuoles سميها هاي الفيتشر microvesicular steatosis, or appear as single large vacuoles inside the cells pushing its nuclei at the periphery. نسميها الماكروفيزيكولار steatosis, or macrovesicular fatty change. In fatty change, the most affected organ with fatty change are the liver liver, heart, renal tubules, and skeletal muscle. This gross appearance of liver affected with fatty chains appear as enlarged or swelled and yellow in color, greasy appearance, and soft in external surface. Mechanism or pathogenesis due to impairment of metabolism of fat, excessive triglyceride accumulation into the cells, and also due to decrease in protein synthesis, including LDL. يصير عندنا defect بالcell injury، يصير عندنا defect in rough endoplasmic reticulum, detachment of or dispersion of ribosomes. راح يصير عندنا decrease in whole protein synthesis including LDL protein أو low density lipoprotein, which is responsible for transportation of fats from the cells into blood. لأنه يصير عندنا decrease in transporter of fats اللي هي ال LDL أو low density lipoprotein، راح تبقى الفاتس stored and accumulated inside the cells، لأن تقل النواقل مالتها، لأن ال LDL هي اللي تنقلها من السيلز into the blood. فإذا قلت يعني صار decrease in LDL راح يصير excessive accumulation of fats inside the cells. The causes, there are different causes: poisons, carbon tetrachloride, phosphorus. مثلا الليفر دائما affect بهذه causes. chronic alcoholism, infection مثل diphtheritic myocarditis, congestive cardiac failure, severe anemia, ischemia, diabetes mellitus, malnutrition, and wasting disease. كلها تعتبر causes of fatty changes. These figures show the typical examples of fatty changes in liver. This is stained with hematoxylin and iosine, routine stains, and this stained with special stain for fats. The cell appear with single large vacuoles fold nearly all the cells and pushing the nuclei at the periphery, and the cells of or hepatocyte appear similar to adipocyte appearance. بين وكأنه adipocyte due to severe يعني single large vacuoles or nearly complete filling with fat vacuoles. فهو هنا تعتبر fatty change لأنه صار عندنا قلنا لأنه الفاتي change هو abnormal fat storage in non-stored cells, in non-normally stored cells. إنه normally الفاتس هو stored in adipocyte، الاديبوس tissue، adipose cells، بينما إذا بقوا accumulated overloaded يعني cells of other tissue مثل هنا عندنا بالهيباتوسايت أو في liver parenchyma نسميها الحالة fatty change or steatosis or fatty degeneration. Here special stain for fats اللي يسمي oil red O appear with red or orange color of fat vacuoles بينما هنا لأنه صار به dissolving during the tissue processing بال H&E إنه يذوب بالكهول يعني tissue processing. بينما هنا السبيشال ستين مصابها الفاتس and appear completely full the hepatocyte. Also other example of hepatocyte in liver parenchyma appear with single large vacuoles full the nearly complete the cytoplasm of hepatocyte and pushing the nuclei at the periphery, similar to adipocyte in appearance. Also this change induce or exhibit accumulation and infiltration of a large number of neutrophils inducing acute inflammation or acute hepatitis. Also there are accumulation of hyaline material. Hyaline material refers to or called Mallory hyaline. دائما هاي البوديز or الهايلاين masses نشوفها associated with fatty change in alcoholic fatty change. alcoholic fatty change دائما نشوف هاي المالوري هايلاين bodies نشوفها accumulated in liver parenchyma. هايلاين change خليها على غير المحاضرة الجاية إن شاء الله. شباب بس أقولكم يعني اللي الثيوري اللي حكيته في هاي المحاضرة، الثيوري هو ما مطالبين به يعني جزء البراكتيك، بالعملي مطالبين only بالفيجرز، يعني either microscopic or gross. الامتحان مالتكم راح يكون عبارة عن سلايدات بارتين، جزء سلايدات يعني بالميكروسكوبس مثل ما امتحنتوا بالبيولوجي بالهيستولوجي، والجزء الثاني هو عبارة عن داتا شو. الداتا شو بالنسبة للداتا شو يعني باوربوينت، ممكن انه يشمل سلايدات مايكروسكوبيك مثل هاي الصورة اللي تشوفونها قدامكم، او سلايد يعني gross appearance for certain case، يعني هاي الحالات اللي راح ناخذها ان شاء الله بهاي المحاضرة والمحاضرات الجاية. فالثيوري بالعملي يعني ما يجيكم سؤال دايركت ثيوري، فقط تجيكم سلايدات مايكروسكوبيك او ماكروسكوبيك او gross، وتجي الكويستشنز عليها، يعني الكويستشن حول الفقرات اللي راح تشوفونها سواء بالسلايدات او بالبارت الداتا شو. الا انه بالمحاضرات الاولى اللي اخذتوها بالهيستوتكنيك ممكن انه يجيكم سؤال ثيوري، بينما بالعملي هو كله يجيكم عبارة عن فقرات ماكروسكوبيك او مايكروسكوبيك وتجي عليها الكويستشنز. والسلام عليكم ورحمة الله وبركاته.